Welcome back, everybody. Thank you very much for being accommodating about my being sick today. I appreciate your coming virtually for the day. Try not to make a habit of it. I need to remind you, as always, that this class is recorded and live streamed. The previous week's ones should be up and available, so I don't know if anybody's tried looking at them, but they've at least been trimmed to get the dead air out and are available to you for review if that's helpful. The slide decks are also always available to you. We try to remember to put them up as PDFs as well as PowerPoints for people who don't like it. So last time we were talking about energy functionals and things like that. We didn't get to talking about Computational Python, so today we're going to spend the whole class talking about Computational Python. I started out by giving you a chance to talk about project ideas, summarize a couple of results which we skipped over a little bit fast last time. And I want to talk a little bit about how to use Twedit, because the model editor that we have is pretty powerful. But it's got so many features that it's easy to lose track of them. And then we'll talk about Python, what are called Steppables in Computational, which have 4 main sections. We'll talk about those. And then we will apply those to some simple simulations. Assuming Giuliano shows up, he'll be giving a little demo. And then we'll talk about how to do plotting and display in Computational, because when you start building simulations, it's nice to be able to see as the simulation is running what's going on, as well as having pictures. And so I hope we'll do some more exercises for that. And then if we have time at the very end of the class, Hayden would like to talk a little bit about project ideas. In particular, if you're going to put something up on NanoHub, you would need to think about what it would be like from a user's point of view. And so he's going to talk a little bit about that. And I'll warn you in advance that there are a couple of things that we're going to show today that are fairly new in Computational. And so those will be available to people running local copies, but not to people on the NanoHub version. We're trying to get the NanoHub version updated. So to begin with, I guess I'd like to open the floor to people if people would like to present project ideas.
So I was actually, these slides are actually more for kind of next week or when I was mostly doing it, but I'll just give it now. Is kind of, so what I'm hoping to do is, so my background is in evolutionary robotics. So, and I'll probably explain a little bit about what that is. And basically what I was hoping to do is to be able to kind of integrate that in a way with what we do in Computational. Computational is really cool, but it would be also really cool to try and see a marriage of these two ideas that you can kind of play around with. But let's see, we'll see how that pans out. So first I'll give a kind of broad motivation for what I was thinking about doing is so that you have all these processes, right? You have regeneration, development, you have things like autopoiesis in cells, or this kind of self-maintaining, self-organizing thing that seems to be very unique of life forms, right? So you have things that, and these all kind of involve these large or small scale cellular collectives that kind of form together and exhibit this incredibly nuanced and interesting behavior, whether it's regenerating a wound or starting from an embryo and being able to grow into an adult life form. All these things are like really, really complicated and involve many, many different types of collective action and collective behavior. So when we're thinking about how we want to start to begin to understand these, a lot of my training in modeling kind of puts this emphasis on starting from the most minimal structure, starting from the most simple kind of units, the simple ideas, and then building up from there. Much more of a kind of like integrative approach. You tack things on as you go, but you first try to make sure and constantly sanity checking, like, this minimal model, is this good enough? Is it doing what we actually think it's doing? So yeah, just kind of going that. So for my project, what I really wanted to do is basically, since all of these different kinds of really interesting behaviors involve these cellular collectives that first organize together, well, if you think about what an individual cell has to do, is it has to be able to get somewhere. It has to be able to go to a location to begin with. So it needs to involve something what roboticists like to call locomotion, or what more of the biologically kind of the biological perspective kind of sees more as cell migration, cell movement, right? But what we have, one of the first things that we say, so if we ask, okay, cell movement, maybe that's something we want to model or something we want to study, where do you begin, right? You know, if we think about one of the paper that we read at the beginning of the class, it kind of it carves out modeling as this kind of building process where you first start from the qualitative aspect of the model, and then you get deeper as you go along. So thinking about it, you know, one of the first things I was thinking is, what type of cell movement are we talking about here? So I'm going to, you know, paint a really broad brush. There's lots of different ways that cells and other, even cell collectives, locomote, move, migrate, whatever you want to call it. But I kind of saw, or in the literature, I saw two major groups, at least in the more simplified, in the more kind of grassroots movement, if you want to think about it as that, is this kind of, so ciliates, for an example, are really common. So that's everything that you see on the right-hand side. So you get a really interesting example is a paramecia. So it uses thousands of the little cilia that are all across its body, and they beat in order to propel the organism throughout some sort of, you know, it could be water, some sort of media, right? And then there's this other one called amoeboid movement, which is and it's called that just because, you know, amoebas exhibit this characteristic kind of crawling, right? So for my project, I simply, you know, because when you think about, okay, what do I need to do in order to model this? Trying to incorporate all of the, and cilia are very nuanced biomechanically, and it's a lot of work to be able to understand that in a kind of rigorous way, where at least I know, you know, in my mind, I was just thinking, okay, you know, maybe I wouldn't have to do as much work. Now, you know, maybe in practice, we'll find that that's not as simple, but it never is. Then I just had a really quick video demonstrating amoeboid movement. I think I could just play it. Yeah. The music is a little strange, but that's all. So it's just this characteristic crawling behavior. So, you know, when we're thinking about in the literature, how does it work? You know, so we can see that it uses this thing called I think it's a pseudopodia. James, you can correct me if I'm wrong, but I think that's what it's called, where it stretches out and it basically then it adheres to the surface and it pulls itself very slowly. So it's kind of this crawling motion. But I know I was reading I found myself one day reading these like really old, like I think it was Herbert Jennings, who like used to do these really interesting behavioral kind of characterizations of amoeba and paramecia. And it's so fun because at that time, biology was drawing pictures of the cells that you saw under the microscope. And I was reading a lot of this. And so actually paramecia, sometimes apparently they exhibit different types of locomotion depending on their kind of quote unquote state, right? So if they're more hungry, I've heard that instead of just this simple crawling, they tend to get this kind of galloping motion or this much more quick kind of locomoting, you know, but that's not something that we're going to be able to capture, or at least at this first stage of the model would be able to capture. So yeah, amoeboid movement, that's basically what I'm looking at. So I guess I'll say a little bit, because I know that I'm coming from a different background than I'm sure a lot of other people are, but I'll say a little bit about the modeling methodology that a lot of my background uses, which for those who are unfamiliar, it's Randy Beer and Eduardo Izquierdo's computational neuroethology lab. It's just kind of the way that we perceive modeling and as it relates to adaptive behavior in an organism. So really quickly, all that means, and when I say like, oh, my background's in evolutionary robotics, this is what I mean, is that our first step, you know, we choose our phenomena. We have that amoeba locomoting behavior that we saw. We're saying, okay, I want to model that. So in some way, as we further classify this into a mathematical model, and we further like try to understand the way that it relates to the biology, we first build that. That's that first step right here. Then, you know, when we finally do that, when we put a controller or some sort of network within some sort of structure that then does the behavior that we're interested in, we then say, okay, well, how do we get the parameters for this structure, right? So evolutionary robotics says that we evolve solutions to this parameter or to the parameters. So we say that we train it in order to do a certain task. Then, this task can not only be a training of the kind of controller's parameters, but it could also be it can also be a selection of the kind of structure in which the organism forms. So Carl Sims, that's what this bottom image is right here. Carl Sims has this really interesting work that he published a while ago now, but he basically, it's just like he had a bunch of robots competing in order to grab something, and they would be able to grow different arms and different other things to kind of gain advantages. And it was very interesting. It was a very interesting kind of solution space to the task, and it was very cool. But that also means that we understand, in some way that means we need a controller. So we need something that's doing the kind of that's processing the information, doing something, right? And that's what you see on the bottom left. Then, it just kind of follows this circle. This is the kind of methodology that it is. This is a little bit more on evolution, kind of just added context. But yeah, so basically the first step of the project, which I'm doing right now, is just embedding that controller in the moving organism. No evolution yet, just trying to get it working, right? And that's really it. That's really it. I more plan to do this later in the semester, but yeah, that's all. I kind of wanted to throw some slides together because, if anybody's interested in the project, please feel free to reach out. Even if you're not. So, let's say, for an example, you're worried like, oh, I don't know anything about robotics, or I don't know anything about all of this. Like, it's really fun to kind of learn and add an added thing. And, I'd make sure that at least, I don't know. That's all. I just wanted to have something to show you guys just in case you could see a different perspective on things. But yeah, that's all.
That's great. Anybody have any questions or ideas? I think it's a great project idea. There are a lot of different things going on in that. One of them is the difference between the mechanical way in which cells move, which you talked about, swimming versus attachment versus other kind. There are other kinds of cell movement as well, and to some extent, some of that has to do with what's going on inside of the cells, which we don't typically model here. Although one could model some of that, or more of a question about if the cell moves in a particular way, what is the outcome? And so, in CompuCell, in particular, we can have the cell move by random fluctuations of its membrane. We can build models of cells crawling with pseudopodia and lamellipodia where there's more detail. I think I showed the very first lecture, I showed a simulation that looks like that. We can build models where the cells actually do make adhesion junctions to the substrate, which is more like what happens when cells really crawl. Or we could do something Boyd's-like, where we simply say there is a force on the cell. We don't say where it comes from. We just assert that there is a force, apply the force, and the cell moves. So one question is the level of detail with which you want to model the particular mechanics of how the cell moves. The second question is, where does the cell decide to move? How much of the cell's energy budget does it use in movement versus doing other things? What kind of control is there on those things? And then you have to make a decision about what kind of the key thing is what kind of information does the cell have? And one very fundamental principle in cellular biology is that cells have access to their internal state, and they have access to local information. But unlike us, we have sound. Well, of course, at some level, vision is photons coming to our eye. However, they're coming over a long distance, and so we think of that as a long-range interaction, not as a short-range interaction where it's just the photon striking the retina. Similarly, sound, of course, again, it's a mechanical vibration in our ear, but the source of the sound can be quite far away. At the cellular level, there are much more limited opportunities for long-range information. So cells really do sense their local environment. They sense their local chemical environment, their local mechanical environment. That's thermal environment, maybe light, but basically the cell's confined to what's within its boundaries, as far, or on the surface, as far as information goes. And that
Can
limited.
I ask there something? Because
Go ahead,
when you have in the human body hormones, they come from different organs and go to other organs. So this is kind of a long range signaling that you have in human bodies.
So hormones are secreted generally in the blood and then transported in blood, and cells respond to the chemical concentrations in the blood. And it's certainly true that you have long-range transport through blood. And so that provides a background set of information that the cells have. So the fact that there is long-range fluid transport in the body adds a level of essentially short-circuiting, which means that there's certain global information that's present. What's a little bit more difficult to get is spatial information that's also long range.
I see it.
But absolutely, that's true. For a cell, a bacterium in the environment, the temperature of the environment that it's sitting in comes from a long distance away in principle. Nevertheless, the response of the cell is to its local levels. But I think that that's right. The place where things get more complicated really in the body is the nervous system, because there you do have long-range transmission of information, and it's spatially and temporally organized in a way that's not true for hormones. And so if you're dealing with innervation by neurons, then all of the rules that I've just stated essentially don't apply or have to be reconsidered. Mm-hmm. And so it's not that so I don't want to say that you never have long-range transmission of information. Nevertheless, each cell itself only can respond to the information that it sees locally. In other words, if the cell is going to respond to a neuronal signal, it has to be in contact with that neuron. If the cell is going to respond to hormone levels, then that hormone has to be transported in the blood or lymph and then diffused through the tissue to get to the cell. And so the key thing is that there are no roadmaps. There's no information for the cell about anything except its local environment. And that winds up being making things like development rather complicated, because we don't have the ability to say, this cell knows that it's in the liver. This cell knows that it's at this position, because the cell only has local information. And so that means that design is a harder problem in biology than it would be for an engineered system. But you're absolutely right. There certainly are things that do get long-range transport. So the key thing is not to say that that you never have long-range signals. The question is to say, in the particular system you're looking at, what are the signals that are available? Because it's very easy to say, we want to know, if you say that the cell there's a food source here, and the cell is going to go and move towards the food source, then the question you have to ask immediately is, how does the cell know where the food source is? How does the information about the food source get to the cell? Is it diffusion of a chemical, in which case you have to ask, how far does that chemical diffuse? Is it something else? And so the key thing is that the cell only has certain kinds of sensory behaviors. And so as your inputs for your control network, you're limited, because you can't ask the cell to have as an input something that isn't available to it. And so that becomes then a challenge. And then you have to ask, what are the outputs that the cell can have? And again, there, certainly movement of the cell is one possibility. In CompuCell, there's a bit of a separation between random movement, what we call intrinsic cell motility, which is random cell diffuses, and directed movement, which can either be something we impose or something that emerges from other interactions. And so those can be controlled. And so I think putting a cell, even just a single CompuCell cell, into an environment and asserting it can move in a particular way, and then ask the question, given the information available to it and some control network, how does it move? Can be a very interesting problem. One thing that we don't have in CompuCell or PhysiCell, but is essential in life in general, is metabolism. In reality, to stay alive, you need energy. If you run out of energy, you die. Or maybe if you're bacterium, you become a cyst, or a spore. But effectively, you need energy to be alive. And so, and to move takes energy as well. In CompuCell or PhysiCell, fundamentally, movement is a free action. And that's not true in reality. And so you have to assign a cost to that movement. And the way you do that cost assignment winds up biasing the results quite a bit. So I think there are a whole series of really interesting questions that you can ask here. You bring up the question of growing an organism. And that's certainly something one can do too. And there's some beautiful old simulations, for example, by Pauline Hogeveg using this methodology, growing organisms. The problem always with those kind of evolutionary systems is you have to have some selection criteria. You had your cycle where you were doing genetic algorithms in your slide. And the question is, what are your criteria for success or failure? If you have an outside criterion like the organism with the most cell types, then the system evolves to have more cell types, which is sort of boring. System with the highest entropy, it will do that. If you actually have something where your organisms compete in an environment for a scarce resource, then you can have situations in which you evolve things you didn't know would happen. But you have to be a little bit careful to set those systems up so that the competition is really between the agents, so that you can actually evolve interesting things.
What I found too is what makes for really interesting stuff is, I think it's called Pareto or something optimization, or using a fitness function that balances multiple needs, basically. I know some work that I did in my lab was, we did something like that for a very, very small robot. And it's really, when you take a task just from, because most tasks in evolutionary robotics are just like walk or get from point A to point B, or from like what you said, or optimize some global thing, right? But I find that it's really interesting, and you start to get really, really interesting results when the organism or the agent has to somehow balance a number of different things, like they would in real life. Yeah, it's super cool.
Right,
So,
bacteria do that. It's a very sorry, Omar, go ahead.
Can you say again what's the Pareto distribution you said, or that you used?
Pareto optimal problems.
Okay, thank you.
So where you have multiple constraints. And this is actually something that we're doing inside of CompuCell all the time, because we write down multiple constraints. You can't solve all of them at once. And so the configuration and evolution, the dynamics that you get are a compromise between all of the conflicting constraints on the problem. And so, in fact, we are doing, a kind of stochastic Pareto optimization in the way we move cells. We don't usually think about it that way because we step back and look at it from a bit further away. We don't look at in the micro scale, but that's actually how the system works. Again, something you'll hear me repeat an endless number of times is that biological evolution is not optimization. The moment you can write down an optimization function, you are not going to get the variety of results and the serendipity and emergence that you get in real evolution. Now, that doesn't mean that from an engineering point of view, evolving robots that move as fast as possible with as little energy as possible isn't a very interesting problem. But it's a little bit different from the question of, if I throw agents down in an environment and let them compete with each other, what happens? Because in one case, I know what I want as my outcome. In the other case, something will happen, but there's no guarantee that the agents will be maximally fast, maximally efficient, or anything else, because the decisions that they will make, which will determine their fitness, depend on the other agents in the system. And so the optimization problem is changing with time and is evolving with the system, whereas in another case, it's a static imposed problem. And so that makes it a fundamentally very different problem. But there are lots of great problems in evolutionary optimization that you could do very well with something like Computational. So I'd be happy to talk about that more. Does anybody else have comments, or does anybody else want to present today? That was great, Gabriel. Thank you. JH, do you want to go ahead?
Let me share my screen. Yep. Unfortunately, can you see my screen? Yeah. Unfortunately, I didn't prepare any slides like Gabriel did, but I just wanted to introduce our team, CC3D, with Jonathan and Elmer. CC3D came from Morpheus because we were gonna translate Morpheus code to Computational. Morpheus to Matrix to Trinity. Yeah, I think it's Elmer's idea. I like the name. The project, the temporary title is In Silico Model of Organoids from Human Pluripotent Stem Cells. It's based on the Libby et al. 2019. The paper is about based on human organoids, which is many biological experiments are based on animal model organisms, such as mice or rats, and they are a good model organism, but different from humans. So these human organoids are valuable tools to study human cells in a human-specific context. So it is a valuable tool, but still it takes months and a lot of effort to create organoids in vitro like this. Before that, organoids, just so you know, organoids is a kind of spontaneous aggregation of cells in 3D. So, so it's kind of, organ if you grow it well. So it's a good in vitro model to study human-specific aspects. Anyway, so it takes a lot of time and effort. So it will be great if we can simulate the experimental conditions in advance to that wet lab research. This kind of in silico model would predict organoid cellular organization and dynamics. So we plan to replicate the computational model in the paper, which use Morpheus and machine learning optimization. But first of all, we want to translate Morpheus to Computacell first, and then like if we can dream high and we have some time left, then we want to try machine learning component as well.
So maybe the next time, maybe not next week, but 2 weeks, you could pull some of the images and basic information from that Libby paper.
Mm-hmm.
Walk us through a little bit about what they do.
Sure.
So I mean, this is definitely something that I've wanted to see for a long time. The idea of being able to design. At the moment, you could use a tissue printer and throw cells down in a dish, but what they do afterwards is up to the cells.
Mm-hmm.
You can't really design the outcome. And so the idea of trying to be able to design experimental conditions, initial conditions, and control conditions that would give you a particular structural or functional outcome for engineered tissue is something that people have wanted to do for a long time and never really been able to do. And so I think that working in that area would be very exciting. And certainly anything that can be done in Morpheus, you should be able to do in Computational. So at least to that extent, it should be manageable. I'm not sure with the machine learning, are they running, for example, a neural network inside the individual cells, or are they using a neural network to do optimization on the system that they're controlling?
I think Elmar could answer this question.
Yes, I quickly can chime in. So the model itself is in Morpheus, but the machine learning is actually external that they do. And it's kind of, they figure out the parameter settings that they have to do. It was on the weekend when we did the whole paper. So the one setting that I had was kind of like a bullseye that you see on the right-hand side. And then you have as well these small islands, and these were the two settings. And I think it was one cell type and one matrix, or two different cell types. I really don't remember now, but they had different conditions, and then a different probability that either the bullseye was reached or the small island model was reached. And they used machine learning to find the right parameters. So it was kind of, if we do the parameter settings in the beginning like this, then we reach to such an amount the bullseye, and if we do the parameter setting like this, then to this and this probability we will have the small island that show up. So that was kind of the story, and they used about 3 machine learning steps in combination. So I think we really have for next presentation to show this in detail, but it was externally done in actually it's MATLAB code that runs on Octavian. So we'll look into it as well. We just downloaded everything and sort of what that it would be possible, but we have not yet nailed in and really took it apart.
Okay. I mean, either way is possible. I mean, in principle, you could. It should be possible to load PyTorch inside of Computational. Would be some work, but in principle, you could do it.
Okay.
It's probably easier to run Computational inside of PyTorch. But I mean, Gabriel was showing us over the weekend, he has a little neural network solver that he wrote in Python, which is a class, and that can be loaded into Computational.
Yes, I understand that I can in Python, in most program in Computational run any Python code, but it's not deep learning. That they did some deep learning. There was some learning from an image, but there were 3 different parts, and not all of them were deep learning parts. But there were still machine learning parts. So I think we really have to separate this out and try to get it more clear what are the parts and where they are. But I know that in this original paper, they run everything of that externally. The Morpheus model doesn't have this integrated, but we surely can try to do that. That's
but
a
no,
further
but it's
challenge.
just it's not that one is better than the other. It's just technically the issues you face are a little bit different, but they're both possible. So why don't we try to set up a meeting with the four of us, where we Giuliano maybe, and walk through the paper together and look at it and think about what's needed to do that. So people seem to be interested in evolution this year, which is nice.
It's great, right?
Other comments or questions from the community? Okay. Does anybody else want to present today while we've got the chance? So even if you're not presenting, just for the rest of the group, how are people doing in terms of thinking about projects? Let's see, we have, who hasn't talked? Hayden, Logan, Ibrahim. Or let's just go down the list. Hayden.
That is gonna get me every time. I'm not used to being in a room with another Hayden.
Yeah, our group has decided on Ibrahim's angelfish cells idea. He would probably be a better person to talk to about that. I don't know if he's prepared for that or not, though.
Well, you don't have to present, just a few words. It's fine.
Well, yeah, I mean, we sort of just met today, a couple hours ago, and decided to go in that direction. So
is this fish stripe patterning? Is this coat patterning in fish that you're interested in?
I believe we're looking at retinal cells tentatively.
Because it happens that Alejandra Volkaning at Purdue does stripe patterns in fish on the body stripe patterns on fish. And there's a very simple model that was a cover story in Nature some years ago of the patterning on angelfish skin. And that's a model. I drafted a toy version of that model many years ago in Computational and never did much with it. And she called me the other day and said, "Is anybody doing that kind of modeling in Computational?" So if that were the aspect of angelfish patterning you were interested in, we actually might have an expert who was interested in talking to you about it. If it's in the retina, that's fine too. One of the things, the thing that was revolutionary with angelfish was something that was completely obvious, could have been noticed at any time for thousands of years, but wasn't noticed until 1998 or something of this kind, which was that as the fish grow, the stripes move. With leopards, as the animal gets bigger, the spots get bigger, but the pattern is frozen. There's an early phase of the embryo development where the pattern is laid down. And then as the animal gets bigger, the pattern stretches. But there's some rearrangement as cells die. But basically, the pattern is fixed. Butterflies, the pattern is fixed. In fish, the pattern is dynamic. As the fish gets bigger, the stripes stay the same width. And you get more stripes. And so nobody had taken photos of the same fish over years to see the fish gradually get bigger and notice that the stripes actually move on the fish.
Yeah, that does sound like an interesting problem that we could talk about at our next group meeting.
And so if you look up in Nature, just say angelfish stripes, you'll find the experimental paper and the paired simulation paper that went with it. But in fish, the colors are given by, there are a couple of cell types. There's a pigmented cell, there's a yellow color, sometimes there's a blue color, and then there are feedback loops, local and feedback loops, or slightly longer range feedback loops that say yellow doesn't want to be next to black, black wants to be next to other black, blue wants to be next to yellow but not to black, et cetera, et cetera. And those kind of signaling rules then can give rise to a whole variety of different code patterns. And then you can also do these kinds of evolutionary things where you say, how strong is this signal? How long the range is this signal? How many of each kind of cell do I have? And you can evolve all sorts of different stripes, spots and stripes, stripes and spots, and so on. But I'm sure that the retinal patterning, which I'm not familiar with, probably has some of the same issues. In fish and in amphibians, one of the things that happens is that the retinas grow continually as the animal gets bigger. Most fish, not all fish, but most fish grow continually. Humans and mammals, typically, we reach a size, we go through adolescence, puberty, and we stop growing. Most plants don't stop growing. Most fish and many amphibians don't stop growing. They get continually larger. And actually, how animals stop, how organs stop growing is actually a bit of a mystery. How does your right leg know to stop at the same size as your left leg? There's no signal back and forth between them. That's actually been a major topic of biological research in the past 25, 30 years. And still not completely understood. But in the nervous system, our somatotopic maps in the brain, how the retina maps into the brain, is fixed in childhood. And if you damage the retina, you can't recover that. Those are fixed. There's a period of neuroplasticity that then ends. Whereas in amphibians, the retina gets bigger, the eye and the retina keep getting bigger as long as the animal's alive. And it keeps forming new retinal projections to the brain. The brain keeps rewiring, and the retina keeps rewiring as well. And that exactly how that plasticity works, at least the last time I looked, which was some years ago, was really not understood. So there are lots of great problems there. Which one in particular you're thinking about? So look forward to seeing more on that. And again, I can meet with you individually, but at some point it would be good to present it to the class as a whole, just so we know what everybody's on the same page, has idea. Great. Okay, thank you, Hayden Ibrahim. So let's see. So Gabriel.
Me and Logan could present next.
Okay, go ahead, please.
Put my camera on. So we are doing quorum sensing, and we do not have any slides presented, but I was thinking I would do a little whiteboard if this will work for me. Can you guys see my whiteboard? Or you know what, maybe I could just share my, oh, here. Can you see this now? So what we were planning on doing is we were gonna kind of simulate a population of bacterial cells, and in quorum sensing, bacterial cells will release these little molecules that are metabolically cheap to produce, easy to make, called autoinducers. And when the bacteria sense a sufficient density of these autoinducers, they know they're surrounded by bacteria of a similar phenotype, and that's when they'll begin working as a collective. So that's kind of quorum sensing, is this phenomena where these cheap molecules, autoinducers, will interact with the genome and they'll start this collective behavior. And in Logan's lab, they work with a drug that is meant to inhibit this behavior. And the way this can be measured is there are a few different ways. So we're still kind of talking about exactly how we would want to measure it. But what we came to last time is what will happen when these bacterial cells realize they're in a collective is they'll start releasing more metabolically expensive material that will help kind of, so this is my food, that help their neighbors grow. They'll like help each other create this colony of collective. So the drug works by stopping the quorum sensing reaction, which will lead to these neighbor cells not putting out their friendly but expensive to make nutrients, thereby lowering the fitness of the whole group instead of just the fitness of a single cell. And then the other aspect to our model is a few different phenotypes that exist. So maybe Logan could talk more about the different kinds of phenotypes. I just know that there are honest cells and cheater cells. So the honest cells will hear the autoinducers and they produce the food. The cheaters hear the autoinducers and they take up the food, so they enjoy the group benefits, but they don't produce any of the food molecules because they're, like I said, metabolically expensive. They're hard to produce. So these cheaters take in the group benefits and they know they don't produce them. So we wanted to look at populations of bacterial cells with different proportions of each phenotype. So sometimes a lot of honest, sometimes a lot of cheaters. And then when they're in a sufficient density, they'll start producing these food molecules that kind of make, help each other thrive. And then we were going to test the effect of adding in the drug that will stop the quorum sensing. There's definitely a good amount of models that look into different aspects of this relationship, but as far as like a drug-centered model, I think we would be doing something rather novel. And then just to look at the chats, yes, Giuliano, I believe they do change phenotype, but that's definitely a question that Logan can answer.
Yeah, so they can evolve in either direction. It's more typical, as far as we know, for like a quorum sensing cell to spontaneously evolve into a quorum blind cell that won't produce any of the public goods that Nick was talking about. It's rarer for cheaters to go the other way because it is, like he was mentioning, it's really metabolically expensive to produce those public goods. And if they don't need to, there's not a lot of selective pressure for them to go that other way. So they typically will only go from contributing to the public good to being cheaters, not the other way. And that was something we talked about modeling as well.
So could you tell me just a few more words about the public good? So are you releasing something that, for example, releases a nutrient from the environment that would normally be sequestered, or something that protects against a toxin in the environment, or something? Because if all I'm doing is taking food in and producing something which is food, which is expensive, I lose. Everybody loses. And so I'm trying to understand what the nature of the public good is. I know there are a lot of different situations, and so I'm just trying to understand which of those you're considering.
So the primary one I had in mind for this was siderophore excretion. So for people that aren't familiar with that those are enzymes that allow them to sequester iron. And those are typically really important in virulent situations where they're in an iron-rich organism. But without siderophores, the bacteria cannot process iron. So it's not necessarily a food thing. It's more of like gathering, like akin to like us taking our vitamins. It's them gathering things that are useful for their nominal metabolism.
Right. So there's a limiting resource.
Mm-hmm.
Okay, so in this case, you have a metabolic resource which is available, which would be some source of glucose, effectively, and then there's a second limiting resource, which would be iron. And you can survive without the iron, or at low level of iron, but having additional iron means that you grow faster, for example.
Mm-hmm.
Is that? But you need some threshold of the limiting of the secondary product to be able to then take advantage of that. Okay,
Mm-hmm.
that helps a lot. That's a great model system. but it's attractive also because it's not so complicated. And so you'd be a cheater basically would say that you have some threshold for switching on the quorum sensing behavior, and by cheating you move your threshold above any level that you're likely to see. Is that what happens essentially?
It can happen a few different ways. The primary way it happens is there's actually a regulator, a transcriptional regulator, and they typically get point mutations in that regulator, so they become like unable to activate the quorum sensing regulon. But a lot of the quorum sensing regulon is co-regulated by other things. So the quorum specific ones are those public goods. So typically those are the genes that are primarily affected by the cheating phenotype.
That's a great example. I think I told you before that one of the first things I did when I got here was organize a workshop with Clay and Eve. It was when Eve was still here, on collective phenomena in single-celled organisms. It's been like 2004, maybe 2006. And we had some very famous speaker who was speaking on quorum sensing. And she actually was saying that quorum sensing is really not a real thing. It's really more of an epiphenomenon, because all the cells are simply sensing the waste products of the other cells and responding to it. And another rather famous and now deceased scientist took offense at this and said something rather insulting. Personally insulting. And there was a fist fight. And so we had a melee. It's the first time I'd ever seen a real melee in a scientific meeting. The speaker got angry and threw a punch. And there was this melee about this discussion of quorum sensing, which was, I have to say, it was nice to see so much passion. Whether having it turn into physical violence was the best outcome, I don't know, but it was an unforgettable seminar. Say that.
I wish every seminar was that interesting.
Well, you don't really want to have to call security at every seminar. Anyway. I don't know if they remember that, but I certainly do. Let's see, actually, it's 2004. Anyway, that's great. So I'd love to see somebody with. Again, I'm happy to meet with the two of you together. Go over it. All right. I guess Carmen and Mike.
Yeah, so I'm also in a group with Hayden and Ibrahim with the zebrafish. So I think the original plan was to look at how cells develop in zebrafish, like in response to different chemicals. And we had planned to ask a couple of questions after class, if that was all right, just because I know you had said that it was zebrafish as a whole are a little bit too large. So we were thinking about narrowing it down. We just wanted to make sure that was okay before continuing.
Okay, I didn't know since I don't have the document in front of me, I don't know who's working with whom. But that sounds fine. As I say, I think zebrafish development has lots of great problems in it. I don't think you could model the development of entire zebrafish. But as I say, things like the stripe patterns in zebrafish are a great problem. And fin development's a great problem. Tail development's a great problem. Segmentation in zebrafish is a great problem. There are lots of vascular development in zebrafish is a great problem. There are lots of really nice neural crest cell migration in zebrafish is a great problem. They're almost an unlimited number. Lateral line formation. So,
awesome. Thank you.
So I guess then we've heard from everybody but Mike, is it?
Yeah, I'm with them as well. The zebrafish group. Yep.
Okay. Okay, did I miss somebody? No. All right, well, that's good then. Thank you, everybody, for talking about their ideas. I'm looking forward to working with you on helping you develop that. So let me come back to the class. And I just want to review a few things that we discussed last time. One of them was this idea that when you're working with Computational, it's important to have an idea about when things go wrong with a simulation, not in terms of things crashing, but in terms of the simulation not behaving the way you expect. One of the things to be thinking about is this idea of the balance between fluctuations and the energy functionals that we're using. And typically what you see is that if the fluctuations are too small, you'll see things like all the way on the right, where the cells will get frozen, not move very much, and line up with the cell lattice. If the fluctuations are too big, you'll see things like the second from the left, where the cell's falling apart into individual pixels, or also all the way on the right, where the boundaries between the blue cells are dissolving. Blue cells basically are dissolving into each other. And so when you see things like cells split up into multiple domains, lots of stray voxels, cell-cell boundaries crumpling up, then the fluctuations are too big. If you see cells not moving at all, cells lining up with the lattice directions or having sharp corners, then the fluctuations are too small. And so developing a bit of an intuition about what is reasonable in terms of cell behavior is very helpful when you have a complex model in knowing where you might look to make it work. We didn't go into much detail about what's called the acceptance function. As I mentioned, the dynamics at Computational consists of taking a lattice site, trying to copy that lattice site onto a neighboring lattice site, evaluating what happens to that effective energy. If the move reduces the effective energy, then you'll make the change always. If the move is opposed to the effective energy, if you're trying to push things uphill effectively, you still can make the change, but with a probability that decreases exponentially with the amplitude of the energy change divided by that fluctuation amplitude. And what you can see here schematically is the probability of accepting a change, which is 1 if you're decreasing the energy, and is decaying as you're increasing the energy. If your fluctuation amplitude is small, that would be something like this blue curve here, the probability of acceptance is only effectively non-zero for energies that are changes that are only a little bit against you. If the fluctuation amplitude is bigger, there's a whole spectrum of energy changes that you'll still make, even if they're unfavorable. And so essentially, how much you can jump over a wall effectively is controlled by that T parameter. And so we haven't really dug into that kind of analysis. It's in the slide deck if people are interested. The other thing that we did a little bit in the homework and a little bit in class, but didn't do the full exercise, was to talk more about the effects of cell-cell and cell-medium contact energies on cell-cell behaviors and cell-medium behaviors. And so one of the things that we could look at was if we have cells that are in contact with each other and with the medium in an aggregate, and we change cell-cell interaction energy and cell-medium interaction energy, what happens? We can find that if the cell-medium energy is lower than half the cell-cell contact energy, the cells actually have a lower energy when they're surrounded by medium than with each other, and they'll separate out and spread out. If the cell-medium contact energy is bigger than half the cell-cell contact energy, then the cells will cluster together. The bigger the cell medium contact energy is, the more smooth and rounded the aggregate will be. And so one can play games with those things, trying to decide which of these situations is more realistic biologically. All of these occur in biology. Depends on what the situation is. Similarly, a small cell-cell contact energy leads to rougher or more crumpled intercell boundaries, as we said before. In some situations, the cell boundaries really are pretty rough and interdigitated. In other cases, they're not. And so we really do have to decide which of these is important. Notice that whether the cells disperse or not in the medium depends on the ratio of cell medium and cell-cell contact energies. Whether cell-cell boundaries are smooth or rough depends on the absolute value of cell-cell contact energy. Similarly, whether cell medium boundaries are smooth or rough will depend on cell medium contact energy. And so I can have a situation in which the cells disperse and their boundaries with the medium are rough, as long as the cell-cell energy is bigger than twice the cell medium contact energy. And so in one case, I have the morphology, clustered versus dispersed, controlled by the ratio of two energies. In the case of the smoothness or roughness of the edges, I'm controlled by the absolute value. And so I have two independent ways of controlling the system. So that's what I wanted to summarize from the previous work and go over a few ideas that we talked about a little bit, but didn't drill into. And now I'm going to come back. The rest of this class is really going to be on programming. And I'm going to be talking about Python-based model specification and Computational. As I mentioned before, Computational does a naive time slicing algorithm where it will move the cells around in response to the effective energies for a while. Then it will go in and solve the diffusion equations. Then it will go in and solve the networks inside of the cells. Then it will go in and do other things, perhaps, and then it'll come back and do another step of the cell movement. Those intermediate steps between cell movement are called steppables, and you could write them in C++ if you felt like it, but typically they're written in Python. Things like the diffusion solvers are written in C++, so they're steppables. In general, we'll do them in Python. And you can, in fact, call the steppables more frequently than once per Monte Carlo step, or less frequently than once per Monte Carlo step. But the default is 1-to-1. A steppable is something called a Python class. And if you're not familiar with Python classes, we'll come back to them in a minute. But if you're not familiar with Python classes, there's a little bit of information in that Python cheat sheet that I handed out. You don't really need to know much about Python classes. They're convenient, but you don't really need to know much about them unless you want to. And Gabriel can talk about why they're useful, and Juliana can talk about it. Come back. Before we go there, since we mentioned CC3DML, I want to come back to modeling things with Computational. And as I mentioned, Computational consists of two components, which people have used, Player, which you run models with, visualize with, and Twedit, which is the model editor. And I know some of you probably are using other model editors rather than Twedit. You don't have to write Computational with Twedit. But Twedit does have some features that are pretty convenient. In particular, and I want to draw people's attention to this, Twedit has some features that make model specification, both of the Computational markup language and Python, much more convenient. In particular, if you are running in Twedit, you will see two menus in this top menu bar, one called CC3DML, one called CC3D Python. These are both pull-down menus. I'm going to talk first about the one that's labeled CC3DML. If you're going to use that, you open the Computational CC3DML model specification. You position your cursor somewhere in that code, not inside an existing code block. That's the one thing you have to be careful about. And you pull down the CC3DML menu, and it'll give you a bunch of options. And those options are metadata, plugins, pots, and steppables. And within metadata, you have a choice of debug output frequency, which is how frequently it writes to the console. Whether you want to use parallel execution and some basic control of simulation properties. When you click on one of these options and that data already exists in the markup language model specification, you'll get a pop-up window that looks like this: metadata module already defined, replace, in which case it overwrites it, comment out and add, in which case it saves the old one and gives you a new one, or exit. And this allows you to paste in code, and we'll be using this repeatedly for the rest of the class if you haven't used it already. I'll warn you that the code that you get is not always going to be exactly what you need, so sometimes you're going to have to edit it. And a lot of times the code will give you options. It'll say, if you want to do this, use this, comment this out, and so on. Occasionally, you'll have to do the commenting out a little bit on your own. The ones that are probably most useful are not the metadata one, but the plugins. Plugins will give you a list of all of those different effective energy behavioral functions that you might want to specify. Adhesion, lists of cell types, chemotaxis, enforcement of cell connectivity, contact energies, elasticity plugins, length constraints, secretion, surface area control, volume control. And so these are all available, so you don't have to remember what the syntax is for the code. Steppables control things that are done, not every micro step in the simulation, but every macro step. The ones that exist inside of XML are mostly initializers, like blob initializer or uniform initializer, or field solvers, diffusion solvers. And there's one for reading in configuration files from the outside at the beginning of the simulation, and one to dump them at the end. The POTS option is to control the lattice size, how long the simulation runs, whether it's square, hexagonal, lattice, periodic, and so on. So just background on using Twedit. We'll come back to the equivalent for the Python in a minute. So now let's talk about the Python Steppables. So the first thing to know is that Python Steppables are classes, and you'll see at the heading of a Python Steppable, you'll see from PySteppables import star. That's the basic enabling code that the Computational provides for this. We'll import Computational, and then there'll be a class definition, the name of the steppable, and the inheritance pattern of the steppable. And again, if you're not used to classes, you don't have to worry about the details at the moment. That's all given to you. You don't have to do that yourself. And then you will find 4 or 5 functions inside of the class. There's an init, which is always a bit mysterious if you're not an object-oriented person. And again, usually we can leave the init alone. The one thing that you will want to know about the init is this line frequency equals, and that is how often that function is called in terms of Monte Carlo steps. It really shouldn't be frequency, it should be period. because it's the inverse of the frequency. 1 means that it's called every Monte Carlo step. 10 would mean it's called once every 10 Monte Carlo steps, not 10 times per Monte Carlo. If you want background on classes, you can see the Python cheat sheet. You'll see that there are 3 functions inside of this class after the init: a start function, a step function, and a finish function. And the ones that we're going to deal with mostly will be the start and the step. And these days, there's also the finish function. There are actually 2 of them. One of them is for the simulation running out on its own, and one is what happens if you hit the stop button. So this is actually a little bit of out of date in terms of the code. We don't that often use those finish ones, although if you're running on a cluster, those are quite helpful. So that's the explanation that I just gave. As always, it's Python. You have to remember to indent properly. So code blocks are defined by regions of the same level of indentation. Computational Twedit has buttons in the top. There's a green button that's a block indent. There's a red button that's a block outdent. And so if you have a bunch of code that you need to indent, you can select all of the code and do indent, and it'll make sure everything's lined up properly. You can also use the tab and shift tab to do that. The start function is called once at the beginning of a simulation, and that's where we're going to do additionalizations. The step function is called periodically in units of Monte Carlo steps. And the finish function is normally called once if the simulation runs to its end. When we're running the simulation on the desktop, typically we'll end the simulation by hitting stop. And now there is a function that you can have called when you hit the stop function. Now let's think about the Twedit. Twedit has the CC3D Python option. It's a pull-down menu, like the one we looked at before. This has not everything. There's still some things you have to look up, but almost everything that you would want in terms of the things you will do in Computational is pre-coded in this rather long menu. And you'll see a long list of things that we can do. I'm not going to walk you through all of them here. I do recommend actually doing that, though, just looking at them. A few of these are going to be used a lot. Cell attributes. I don't remember how to specify what the volume of a cell is. I don't remember how to reference the target length of the cell. That's hidden under cell attributes. I want to create a field for displaying the state of a cell. That's extra fields automatic tracking. We'll use that today. I want to create a graph, which is going to plot the volume of the cell versus time. That's scientific plots. I want my cells to absorb a nutrient from the environment. I want them to secrete nutrient into the environment. That's under secretion uptake field intervals. And I want to do something to every cell of a given type, to every voxel in the simulation, to the cells that are adjacent to another cell. Those are all listed under visit, which maybe isn't the most transparent name. So anything that involves iterating over cells, neighbors, voxels is hidden under visit. The exact list of options here will change depending on the specific version of Computational that you're running. All of the ones that I've listed here will be in all of the versions. A few of these things will be new. And so if you're running an older version of Computational, for example, on NanoHub, a few of these options may not be there. And there are always a few new functions added to Computational that haven't found their way into the editor yet. Okay. As a matter of code design, there are some principles. One of them is that you can modularize your code by using multiple classes, multiple steppables. It's perfectly possible to write a computational simulation where everything gets put into single steppable. That works just fine. But if you're going to reuse modules for other purposes, breaking them apart is useful. You might have one module that controls cell behavior, another one that handles data and plotting, another one that controls nutrient secretion, another one that, suppose you were doing radiation therapy, you might have one that applied radiation to the system. Those could be broken up into different classes. The one thing that you do have to remember is that just the way that variables that are local to a function are not available outside of that function. Variables that are local to a class are not available outside of the class. And so one of the things as a matter of programming that you have to get used to is how Computational makes it possible to make information available between classes. One way you could do that is to use globals. There are some inconveniences with that. But we'll come back to that in a bit. For the simulations we're going to be doing immediately, we're going to be using a single steppable, so it's not going to be a problem. Okay. So the next thing we have to think about is how Computational represents cells and how we manipulate them. So a Computational cell is a rather complex object. We typically will reference cells under the handle cell, but in fact, there's nothing special about the name cell. We could assign cell, the reference to the cell to be something with a different name if we wanted to. Each cell has a unique identity. It's associated with an index. And so there's a cell.id, which is the index of that cell. And in general, the cell has a variety of attributes, which are accessed by typing cell.attribute. And if you're use Python a lot, this will be very familiar. If you use C++, the syntax looks a little bit different. If you're not used to object-oriented languages, this may be new. Cells have quite a few attributes. Under that CC3D attributes, cell attributes list, we have a list of most of the attributes. Cell.id, the identifier of the cell, cell.type, the name of the type of the cell, cell.volume, the actual current volume of the cell, target volume, the assigned target volume in that energy constraint function for volume, lambda volume, the strength of the volume constraint, pressure, the pressure of the volume constraint if it exists, surface tension, the surface tension of the surface area constraint if it exists, and so on. Again, you have to look at you can look at the list if you don't remember what's available. Occasionally, there may be something new that's not in the list. So it's possible that there are things that exist that you have to look at. Okay, so that tells us what cells can do. As I mentioned, visit is a very important concept. If we're used to classic procedural languages, loops are thought of as iterating typically over numerical values. In Python, iteration happens over lists. And so you can't say for A equals 1 to 5. You have to actually create a list with the numbers 1, 2, 3, 4 in them, and then pass it to the iterator. That has the advantage that you can iterate over rather complex lists of things like cells, not only over numbers. There are some disadvantages. One, the order in which you do your iteration isn't necessarily determined. And two, you're not allowed to change the list you're iterating over while you're inside of the loop. And so within a loop over cells, you should not be creating or destroying cells. And so that's something that one has to be aware of. If you like, biologically, when we do for cell and cell.cell list, which does something to all cells, we should think of those things happening simultaneously, not sequential. And so if I come down to visit, I'll see visit all cells of a given type, all clusters, all cell lattice pixels, boundary pixels of a cell, which is a useful thing to be able to do, neighbors of a cell, pixels within a cell, and again, another kind of cluster thing, which we haven't talked about. The one that you're going to use the most often will be for cell in self.cell list, which will iterate over every cell in the simulation. And for cell in self.cell list by type, and then a list of the cell types. Remember, in the beginning of the simulation, you specified a list of the cell types. This is where you would use that. The key thing to remember when you're iterating over types is that the type name is always in all capitals. Even if the name of the cell type is actually lowercase, if I create a cell called stem cell and it's lowercase, the type name would be stem cell in uppercase. So why don't we use what we've seen so far? And I'm going to ask everybody to fire up CompuCell and use Twedit Wizard to build a simulation which has two cell types. Start with one initial cell, have the target volume grow at a constant rate, have the cell divide when it reaches a fixed doubling volume. So that'll be our first exercise. Okay, any questions about that simulation? We'll be using this repeatedly as we go through the afternoon. So let's take a minute to do that. And I'll do the exercise too while we're at it. Do people remember this? It was actually it was one of the things we did in the first class, I think. The first or the second lecture was this exercise. So I hit new CC3D project. I gave it a name. I called it Lecture 4 Simulation. I'm not going to change the size of my lattice. I will use blob initializer. Makes it a little easier to do a single cell. Then I'm going to create two cell types. One, add cell 2. Add. Next, no chemical fields. Now contact energy, growth, mitosis. And I didn't ask for anything else, so that's all I'm going to do. Finish. There's my code. I'm now going to run it in player. And it looks like I did something wrong. What did I do wrong? All my cells are disappearing. Oh, I know what I did wrong. What did I forget to do? Juliana? It's a good opportunity to use the markup language editor. I forgot to give the cells a volume. Okay. I was hoping that people would be doing this independently, not watching what I was doing. Let's see where we are. You should probably have the code from lecture 1 or lecture 2 where we did this, but you don't want to reinvent it. And if people get stuck, then please speak up and ask for help. Now you'll notice that I don't have a single cell to start with. So if I want a single cell to start with, I can use my blob initializer. Blob initializer draws a rough circle of cells centered wherever I tell it to. The size of the cells here is 7 by 7. The radius of the blob here is defined as 51. If I make the radius something like 3, let's see what happens. I'll get a single cell. If you make the radius a little less than half of the size of the cells, you should get a single cell. Okay. So again, if we want to do this, reminder, we go to Twedit. In Twedit, we go to CC3D project, new project. Give it a name like temp demo because I'm not going to be using this one. Again, general simulation properties. The only thing I change is blob initializer, uniform initializer. Create two cell types. Cell 1, add. Cell 2, add. Next. No chemical fields, skip. We're going to have contact energy, growth, mitosis, and I forgot to click volume, which is why the cells disappear. I have to click volume. And it might be interesting to click surface. So we can add that later too. Then I hit next, finish, and that's the full simulation. That's all I need. Open in player, see what that gets. I have multiple cells. I go into my code. And I change, now I'm in the new one, that's my new one here. I go into my markup language. I find the initializer, blob initializer, and I change my radius from 51 to 3. Now, I start with a single cell. So if people are having trouble, please speak up so we can help you out. If people have got this, this would be a good time to take a break. So why don't we say, people who've gotten this far, well, let's take a 10-minute break. And for people who need help, we'll stick around during the break and help out, okay? So who needs help getting this working? Or does everybody have it working? I can do another poll if you want.
I have it working, but I forgot to click contact. How do you change that, like when you're already in Twedit?
Okay, that was exactly the perfect question. That's why we went over the Twedit. So if I'm in Twedit and I didn't define contact, suppose I forgot to define contact. I go into Twedit. I go into a space that's not inside of a plugin. So I have to find a space that's not inside of it. I go to CC3DML, plugins, contact, and there it is.
Okay, cool. Thank you.
Great. That's why those code snippets are really useful. Now, if I'd specified different values, if I changed the values of those energies, then I have to remember that. But the basic structure is there. Anybody else have an issue with getting it to work? There was a question about why medium is a type 0, an object of type 0. Well, the basic philosophy of Computational is that you shouldn't get anything for free, but the empty lattice exists, and you have to fill it with something, and so it's filled with medium. And yes, medium is somewhat of an annoyance because medium does not have a lot of the attributes that other cells have. It's also important, Giuliano points out, it says it made a big computational difference when CC3D got started. That's true. It's also important when you're doing parallelization. If you substitute medium with a cell called medium 2, and you make a single huge background cell that you call medium 2, and then you ask the Compu cell to multi-thread, it will crash. And the reason for that is that to parallelize, you have to be able to do lattice updates independently of each other. And if you have a very large cell that fills the whole lattice, then an update in any position in the lattice can affect every other position. You have non-local information. And so it's important that that medium cell actually not can convey information. Otherwise, the parallelization fails. If you want to have the equivalent of medium with a medium 2 cell type, you have to subdivide the medium into lots of little blobs that don't fill the whole lattice. That's a sort of a high sophistication question. It's a good question to ask. Is anybody else having trouble getting the base simulation to work at this point? So we can debug while people are getting their break. Okay, I will see you back in 6 minutes then. We'll start back at 6:50. Okay, so there's a good question. Can I limit the growth of the cells? And the answer is that will be one of the things we're going to do in the next bit of the exercise. So you're a couple of steps ahead of us. Is everybody back? Does everybody have it running now? Let me poll again and make sure that this time everybody has it. Yeah, thumbs up is fine instead of a poll. I have 8 yeses and 10 people in the room. So who are the 2 people who didn't, who haven't answered? So we're going to use this for the next hour to build forward. So it's important that we have this. The first thing that we were going to do, Giuliano, are you there? He may still be on break. Giuliano is going to show you how to create a cell in Python. But let's come back to that if he's on break. So what I give him one more minute. No. Okay, what I'd like to show you now is how to plot things. CompuCell has pretty flexible plotting routines. We can plot time series, histograms, and that's pretty useful if we want to monitor how our simulation is running. And so to create a plot, we're always going to need to do the same 3 things. The first thing we're going to have to do is create a plot window, which will be a pop-up window that will display the plot. The second thing we're going to have to do is define the data series that are going to go into that plot, because we could plot different things in a single plot. And then we're going to have to add data points to that data series. The creation of the plot window and the data series is going to be done typically in the start function. Remember that we have a starts function and a step function inside of our class. And then adding the data points will typically be done in the step function. In principle, we might want to do something else like save the plot in the finish function if we want. And so to create a scientific plot, we have a utility in Twedit to do that. So let's come back to our code here, and we're going to go into the Steppables now. And here we see Python Steppables, class constraint initializer Steppable. And we see a start function and nothing else. And we see a second class called growth Steppable, which has an init and a step function, but no start function. And here we have a growth Steppable, and that has, sorry, the growth Steppable is there. And we have something called mitosis Steppable. It also has an init and a step function. So in this case, the start function and the step function have been separated. That's actually going to be a little bit inconvenient for us today because for scientific plots, you need the definition of the window and the plotting to be done in the same Steppable. And so what we're going to have to do is we're going to have to copy the start function definition into one of the other Steppables. But let's do this. We're going to do a scientific plot, and we're going to use Twedit ++ to do that. And so to begin with, I'm going to ask everybody, does everybody have code that looks like this? Does your code look different? It may not be exactly the same. It should look similar. Hayden says it looks the same. Okay. Can everybody go into their code, find growth Steppable, and simply copy this line 10 where it says def start. Copy that and paste it in here. Before the step function. You don't absolutely have to do this, but it'll make our life a little bit easier. Okay. Everybody able to do that? Give me a thumbs up if you will. Okay, great. Okay, fine. Now I'm going to go into the start function. And I'm going to go to CC3D Python, scientific plots, and I'm going to go to setup. And that will paste in some code. And here, because of my screen constraints, I'm going to have to scroll. It's going to do something called self.add new plot window, which will create the plot. And then there will be add plot, which will create a data series. And we'll come back to the code and I'll show you that in a little bit more detail. But you can follow along with that. So go into start, find a space below the start function, CC3D Python, scientific plots, setup. And this is what the code looks like. Here it's truncated a little bit, but here you see the whole code. Self.add new plot window is a function that creates a new window in Player. What I've assigned here on the left, self.plot window, is a handle to that window. And I could give any name I wanted on the left as long as it's unique. So if I wanted to create 3 plot windows, I could do self.plot_win1, self.plot_win2, and self.plot_win3. So I'm allowed to have 5 windows, 10 windows if I want. One thing that I do have to be aware of is that, and I'm not exactly sure why this is the case, that each window has to have a different title. And so the window title here, I said average volume and surface. That name has to be unique. If I create 3 plot windows, each plot window has to have a different title. Then I can label the x-axis, the y-axis. I can say whether the scales in the x and y direction are linear or logarithmic. I can say if I want grid lines. And there are some additional options which you'd have to look up. Quite a few of them. If I want to add a data series to that plot window, I have to remember it's called plot win if I had more than one, which one it is. And then I give the data series a name. Here I'm calling it M volume. And then I have to say, is it lines or connected lines or dots, the color, and how big it is. And there are some additional options which you could look up if you're interested. You're going to have to remember the reference plot_win. You'll have to remember that. And the name of the data series, MVol in this case. All right. If I want to display a key, because I have multiple data series inside the same plot, I have a configuration option called legend. If I set that to true, the plot will show the names of the variables. If I set it to false, it will hide that. Sometimes it's convenient to have it. Sometimes it gets in the way of seeing what I want to see. Once I have created this block, I then have to add data points. And the data points I will add in the step function. So here I could go to the step function. And I'm going to go to CC3D Python. And by the way, what I'm typing here is not going to be code that I'm going to have that will work. We'll have to modify this code to make it actually do what I want. And I want you to see how to paste it in. I go to scientific plots and I hit add data point. And if I look at what's pasted in, it will add self.plotWin, add data point, and the defaults are mvol and msurface, the 2 series that I added before. If I had a different name for my plot window, I'd have to change it here. If my data series had a different name, I'd have to change it here. And after the data series name, I'll have an X coordinate and a Y coordinate. In this case, I have time as the X coordinate and the value of the cell volume as the Y coordinate. But in principle, I could plot any 2 X and Y values I want. I could plot surface versus volume, for example. There's no reason that the X-axis has to be time. What is important is that the reference to the window, plot win, has to be identical to what I created. And the name of the series, mvol, has to be identical to what I created. Now, I'm not sure if I try to run this, what will happen. It may crash. Let's see. You see here, it's opened a window, which is a plot window labeled average volume and surface. And if I time step it, It's plotting something. What is it plotting? Anybody have a guess of what it's plotting? Look at the code. This is literally the code I'm running. What am I plotting? Cell division. What is the x-axis here? Somebody, Hayden or Gabriel. What's the x-axis? It's labeled, so maybe too small to see, but it's labeled.
It's the Monte Carlo step. But is this actually bouncing between, like, it's not plotting them individually? Is that why it's looking strange?
I'm plotting the volume of the cell. If I look at my code, I put my plot inside of this 4-cell loop. So I'm plotting the volume of each cell. So at each time, I start out with 1 cell, so I have 1. I have then 2 and then 3. It's a little bit more confusing because my style here was lines. If I make my style dots, I think it'll be clearer what's going on. There's the volume of the single cell I created to begin with. It goes down for a minute because the system is equilibrating. Now I've got a growth steppable, so the cell is growing. I see the cell actual volume doesn't grow exactly in line with the target volume. I can plot the target volume as well as the growth actual volume. The cell grows with some fluctuations. And what happened here? The cell divided, and now I see 2 dots because I now have 2 cells. I told it to plot volume and surface. Why am I not seeing the surface? Elmar says much smaller than volume. Did I define the surface energy? When I defined the simulation, I defined the volume energy. I defined the contact energy, but I didn't define the surface energy. So there's no surface. The surface doesn't, objects don't exist. I'd have to tell it to include them. That's you don't get anything for free. So there is no surface to plot at the moment. So this is just a summary of what I've just said. Okay, so now your assignment, a little exercise, is here I've labeled this average volume and surface. I want you to do 2 things. I want you to figure out how to get the surface of the cells. And we've already done that a little bit by hand because I showed how to add a plugin to the XML. And you need to write a little bit of code in this loop that instead of plotting the volume of each cell, the surface area of each cell, will average the volumes and then plot the average, okay? Okay, how's it going, people? So what I did, and you may have done it slightly differently, was the first thing I did was I went into the step function. I created a dummy variable to hold the total volume, called it mvol. Then I summed it up when I stepped through all my cells. And then I moved my plot outside of the loop, and I plotted mvol divided by float of the length of the cell list, which is the number of cells. So that gives me the average. You could definitely do something more Pythonic, like make a list or a NumPy array containing all the volumes, and then use a NumPy average function or mean function to get that. But here I did it in an iterative way. I could do the same thing for the surface area. And then I went into my XML, and where I had plugin for volume, I added plugin name for surface. And to do that, if I didn't remember how to do it, I could go into plugins, surface, and it'll give me the surface plugin with the values pre-specified. In this case, I don't need those specifications, so I can get rid of lines 31 and 32. And the last thing that I had to do is to get things to show up on your screen was to make my dot size bigger, because the default dot size was 1, and that's too small to show up the way I'm doing the screen share. Okay. So then when I run that, this is what I get. Okay, did that work for people? Anybody stuck? I see most people said they're finished with the exercise. If anybody's stuck, please speak up so we can help out. Okay. Seems like everybody got it. If you're shy about speaking up, you can text Giuliano in the chat. He can help. We've always, if you don't have to speak up. If you want help, you could always text Giuliano and he's here to help. Okay. So we actually started out by plotting the volume of the surface area for all the cells at once. So I'm now asking you to do something slightly different, which is very simple. Remember, I said you don't have to have the x-axis be Monte Carlo steps. So now I'd like you to change the plot, maybe add a separate plot is better than getting rid of the one that we have, where the x-axis is the surface area and the y-axis is the volume. And in this case, you could do it for the mean, but it's more interesting when you do it for each cell. So is it clear what I'm asking you to do? So at each time step, you go through the list of all cells, and on the x-axis, plot the surface area of the cell, and the y-axis, the volume. Try that. And this time I'm going to want one of you to show me what they got. If you want to modify the existing plot, that's fine. Or if you want to try figuring out how to do another plot window, that's fine too. Nick, you got it? You want to show us what you got?
Sure. Let me find it. Okay. Do you want me to show you my code or just plot that I ended up getting?
We can see. Let's look at the plot first and then the code.
Okay, well, here's my plot. I named it average volume over surface. It's called average, but it's actually not average. It's done individually. I mean, here's what it's looking like. Pretty chaotic. And then the, oh, but it looks like it may be getting a little larger as it goes on. The way that I did it. So I added a second plot called average volume over surface, where my x-axis was volume. Oh, that's actually backwards. My x-axis should be surface. My y is volume. And then in my step function, so it seems I'm only adding it for the last one actually. So I'm glad I showed you this because I ended up doing it wrong. What I meant to do is inside my loop, I wanted to plot, this is my plot, is the ratio of the cell volume over the cell surface. And then now that the code is correct, maybe we'll see something different.
You'll see more data points.
Oh, wow. Yeah, super interesting. They
So-
almost look like lines in the beginning.
So one thing
Yeah.
that you might want to try is, maybe I wasn't clear, and that's fine, is you're plotting the ratio of the surface to the volume. Why don't we go back to your code and make one little change? Let's see your code again. We don't have to change the labels. But let's actually, instead of plotting the ratio at each time, let's actually plot the x-axis being the surface and the y-axis being the volume. So if you get rid of MCS comma, and then replace the divide by a comma, now we'll see, there you go. You could still leave the names because they don't matter. That's sort of interesting, because now it'll draw. You should see it draw the different values as it runs.
All right.
Okay, so there's one other thing, right? Well, that's not. That looks like that's probably what it really does.
Yeah, I can see. So it's like almost like building a histogram.
Exactly. The other thing is, one small thing is, if we go back to your code, when you create the second plot window at the beginning in the start function, you've given the name plot window and the title the same name. If you look at line 24 and 33, you've created two plot windows with the same name.
Oh, I
and
made
the
this
same
one.
title.
Are you talking
Okay,
about?
so you got right. So the title is different, but the plot window is still has the same handle.
Okay.
And so in line 33, where you say self-plot plot window on the left, I'd call that self-plot window 2. Just add a 2 at the end. And you
So
have to add a 2 there. There we go.
And then would I want to change it in my steppable also?
You'll have to do the same thing.
Okay.
I don't quite know when you have when you create two plot windows with the same handle. I think it'll just have. I think the second will just eliminate the first one, but I can't promise that will happen.
Well, if we look, my average volume and surface wasn't getting any data points, so that makes
Right.
sense.
So let's try that now. See whether that. So this is just a reminder for cell and self.cell list by type, if we want to iterate over cell 1 or cell 2 type. Critical thing that I mentioned, which was important, which was you didn't see, was that we have to specify volume either in XML or in Python, but we can't specify values for both. We still need to have the plugin loaded in the XML, but we can't specify the parameters for the cells in the XML if we're going to change them in the Python. And so by default, if we paste in the volume plugin or the surface plugin, it'll give us values for each cell type. And if we want to change those in the Python, we have to go and comment out or eliminate those and replace them with just a bare reference to the plugin. So what we could do now is add target surface and lambda surface to the cells. And so we could do that if we go back to the code. It may be in a slightly different place in your code. In my code, it's lines 14 and 15. I'm doing my initialization in my start function for cell and self.cell list. That's for every cell. And I'm setting the initial target volume to 25 and the initial lambda volume to 2. Let's just copy those. And I'm going to set target surface to 50 and lambda surface to 2. Suppose that I don't remember the name of the target surface or lambda surface. I go to CC3D Python, cell attributes, surface or target surface, and it'll tell me target surface. If I forget exactly how to type it, I can look it up there. Okay. So why don't people try adding those two lines? What do you think will happen if you add those two lines?
It didn't make a change, but I realized I defined target surface in my XML. So let me make that change and then see.
Okay. Oh, I have a typo. What is my typo? Ah. So when I run it now, the surface area of the cell stays not perfectly aligned, but almost fixed at 50. Before it was fluctuating along with the cell line. If I look at the simulation of my cells themselves, you'll see that the cells I have too much surface area per cell. So the cell surfaces get very wrinkly. As the cells grow. Were people able to get that to work? Anybody else having problems with that? So again, I just added cell.target surface, cell.lambda surface. You might want to comment those out. This was just showing you the principle of doing. So the next thing, and this is probably the last thing we'll cover today, and something we're going to use a lot. Perfect timing. So JH, you didn't miss very much. We were just playing around with plots a little bit more. So, but you came back just when we were going to talk about the next important concept. And the next important concept that we're going to use a lot in Computational is the concept of a cell dictionary. Computational cells are C++ objects, and their attributes are added in C++. And so we can't, by hand, unless we go in and do things in a rather sophisticated way, add an attribute to a cell like cell.myAttribute. To avoid having that problem, Computational comes with cells having a dictionary. Dictionaries are a very, very useful Python concept. They may seem a little bit alien at first if you haven't used them, but most people, I think, have done some Python. And so every cell comes with a dictionary. And you can put in that cell dictionary additional information. And so if I want to say cell has a name, instead of typing cell.name, which might be the most natural thing to do, I can type cell.dict, dictionary key entry name, and then I can say hello. So this dictionary entry could be anything. And I do mean anything. It could be a string, a float, a list, a dictionary, or even a class. Say, why would you want to put in a class? Well, if I wanted to load a neural network into my cell, I'd want to have functions that operated on the neural network that went with it. So then I'd load a neural network class into my cell. And so this is a very powerful concept. I could say, suppose I want the cell to have its history of its volume. I could say cell.dict, quote, volume history equals empty list. And at each time step, I could append to that list the current volume of the cell. And so I can add complex objects to my cells. An advantage of this is that cell.dict is a property of the cell. And that means that that information is available between functions, between classes. It's fundamentally a global property of the simulation. And so that's really a very useful concept. And so we're going to get used to the idea that if there's something specific that you want to add to a cell, you're going to add it in the form of a dictionary entry. When you divide cells, when cells divide, the intrinsic attributes of the cell are copied automatically. Computational has a very useful function called clone parent to child that will also copy all the dictionary entries. So whatever you define as a user will automatically get cloned to the child cells using that clone parent to child attribute. I wasn't going to show that right now, but we'll come back. I don't remember whether that's enabled automatically in division or not. Yes, it is. So line 89 here. will automatically copy in the division anything that we add to our cell through the dictionaries. That's actually a pretty powerful concept. It also happens to be computationally pretty slow because it uses something called Python deep copy. But since you don't divide cells that often, it's not a problem. So anybody have a question about dictionaries? Because this is a pretty fundamental concept. And again, if you've used Python a fair amount, it'll be familiar. If you haven't used it before, it may take a little bit of getting used to. But it means you can add any variable, any list, any structure that you want to the cells, and you can operate on them with whatever the normal methods are for that object type. The other thing to remember is that sometimes you want things that are globals. And globals are possible. You can use the global function in Python. But a more Pythonic way of doing things is to use the fact that there is, in fact, a dictionary for the whole simulation. Unfortunately, the name of that dictionary is not self.dict, which would be the natural thing for it to be. It's self.shared_steppable_variables, and then the dictionary key. But you can add to that global set of that global dictionary anything that you want, parameters, variables that you want to be available between functions within a steppable or between steppables. And that's a reasonable way to do things. Giuliano pointed out that you can actually, within a steppable, create an attribute name that takes on self, in which case all of the properties of a particular steppable are loaded to be available elsewhere. I wouldn't recommend doing that unless you're pretty sophisticated with your Python. Because remembering what that self context means is a little bit tricky. Can I just sort of, as a show of hands, how many people are comfortable using Python dictionaries? Give me a thumbs up if you want. Okay. So the majority of people have used Python dictionaries before. If you haven't used a Python dictionary before, strongly recommend looking at that little Python cheat sheet. But basically, what we are doing with the Python dictionary is we are creating a place to put information, a variable, except that the variable, instead of being x equals something, that variable is attached to the cell, and it's attached with a name that is put in quotation marks. So instead of saying x equals hello, we say cell.dict quote x equals hello. And then anytime we want the value of that, we'll reference it using cell.dict and so on. Why do we need to do it this way? Well, because every cell will have its own name. And so we don't know a priori how many different cells we're going to have. And so we need to be able to flexibly attach the same concept, namely name, to every cell. So that's why we use this kind of stuff. For the moment, if you can just use things like strings and floats or integers. But as I say, it's sometimes very useful to attach a list or a more complex object, which could give me the history of the cell. When I have the cell divide, who were my parents? My parents were ID numbers, were cell index 10 and 15, if I want to keep a lineage trajectory of the cell. So that's a pretty powerful concept. Okay. We're out of time. I was hoping that we could talk about pressure. Pressure in the cell is the difference, effectively the difference between the actual volume and the target volume. The membrane tension in the cell, the difference between the current membrane, the target surface and the actual surface. There's a minus sign involved. And so the pressure and volume that are returned will be minus the ones that you expect. And so the assignment, which will now be a homework assignment, is to plot the pressure and the volume of the cells as a function of time in the simulation. Something that you might want to do is in the step function, you can change the rate at which the step function is called. But sometimes within the step function itself, you want to only do certain things every so many time steps. And a simple way to do that is to use the command if not MCS percent sign and then the frequent, or then the period. Percent is modulo. So MCS percent 10 is the remainder when you divide MCS by 10. So if MCS is 10, 20, 30, MCS percent 10 will return 0. Otherwise, it will return something not 0. Not of that will be 1 every 10 time steps and 0 on those intermediate time steps. And so if not MCS percent interval will invoke this every X interval Monte Carlo steps. And so you can do something like that, where you add the, every so often you add to your data set the additional information that's being asked. So I think that's enough for tonight. We're a little bit over time for that. And so I'm going to stop there. But that will be enough for you to do that little homework exercise. If you're not comfortable doing the little homework exercise based on what we have, that's okay. We'll cover this in more detail next week. And we will come back to it.